So welcome to the BVA Foundation. Is our sponsor for our Demography Today lecture series that is sponsoring our lecture series for 3 years. This is the starting of our second year, and these lecture series are organized in collaboration with LOMPOP Horizon 2020 project and the Spanish National Research Council. The lecture is transmitted live and then is recorded, just in case that someone wants to know beforehand, before we are starting. So we are very lucky today to have here as the first speaker of this lecture series, Cécile Viboud. She is a mathematical epidemiologist in the Division of International Epidemiology and Population Studies at the Fogarty International Center in the US National Institute of Health, where she has been working since 2003. Her research focused on the epidemiology and transmission dynamics of acute viral infections, particularly influenza, and the interface of public health and computational modeling. She has recently become interested in the transmission dynamics of Ebola and MERS, and the potential use of big data for infectious disease surveillance and forecasting. A native of France, she received an engineering degree in biomedical technologies from the University of Lyon in 1998, and a master in public health in 1999, a PhD in biomathematics in 2003 from the Pierre and Marie Curie University in Paris. Actually, our LOMPOP project is a Marie Curie action from the European Union. So welcome to the lecture series. As usual, we have around 1 hour talk, and then we will open the floor for questions. So we will end around 8:30 or so. So whenever you want. Thank you.
So today I wanted to give you an overview of the transmission dynamics and epidemiology of pandemic flu, because this is a subject that has focused a lot of our research in recent years, and a lot of my usual collaborators are here tonight, including Gerardo, Shawal, Nona Simonsen, as well as Don Olson and Peter Sprovenberg and John Paget, for whom it's very difficult to get a picture online, so you should get one. So before starting, I just want to introduce in a few words a project that we've had at NIH at the Fogarty International Center, where I'm at. We're a division interested in computational biology for infectious disease, and in 2001, Lone and I and others started this project called MISIMS, originally an acronym for Multinational Influenza Seasonality Mortality Study, which has then expanded to look not only at mortality, but also epidemiology in general and evolutionary dynamics of influenza. And it's a global research network involving collaborators all around the world. So we've done quite a few studies on transmission dynamic, time trends, age and geography, control strategy, looking at the impact of vaccination, as well as the antigenic and genetic characteristics of the virus and how that affect the populations and the animal-human interface. So this is just a map of some of the data that have been collected through this project over the last 15 years or so in red, and then some of the workshops that we've organized around the world. We've down to 18 now, so we have 1 to 2 a year in different regions of the world, and 150 or so publications on epi and evolution of influenza. And we also have a website, which was here, right, misims.net, with all of our activities and papers online, and some description of our staff. So pandemic flu, just a quick timeline to refresh your memory about the different subtypes. Oh, it moves of its own. The different subtypes that have been circulating over the year. So the first flu virus was isolated in the 1930s, but through reconstruction and archival autopsies, we've been able to identify the virus from the 1918 pandemic, which was an H1N1 virus. And as was discussed earlier this morning, the evolutionary history of that virus is still unknown. 1957 marked the emergence of the H2N2 viruses. Only the surface antigen changed, and the internal genes of the virus were kept from the earlier virus. and then H3N2 in 1968. So as you can see, only one of the surface antigen, the H2 for the hemagglutinin changed, but the neuraminidase, the second antigen, remained the same. And then resurgence of H1N1 in 1957, which is thought to have come from a freezer. So evolutionarily, this virus was also frozen in time, and only people over the age of 20 were affected by it. And then in 2009, we've had the H1N1 pandemic, which was relatively mild, but actually a really good source of data for epidemiologists to try to understand a pandemic dynamic in a modern population. And then since 1997, we've had the constant spread of avian H5N9 and more recently of H7N9, which have kept resurging in Asia and in particular in China. So that's always something to think about as a pandemic threat for next time. And before 1918, it's really unclear what was circulating. There's some serological evidence suggesting that it was an H3, maybe H3N2, H3N8 like virus, but it's actually relatively little evidence for that, and probably little hope of finding material from that time to try to get confirmation. So just to give you a short overview of the impact on some of those pandemics, they're quite different. So 1918, which is considered by many as a catastrophic scenario, an estimate of about 15 million deaths, and some of those estimates are still being refined, right? And we actually are working on those here in the audience. A lot of the deaths, 95%, were in people under 65 years of age, although at the time the life expectancy was, for instance, in the US, only about 47 years, right? So when looking at the age distribution, you have to take this into account. In 1957, we had an impact that was relatively low, about 2 million deaths, and fewer deaths in people under 65, and we're going to look at that in more detail later on. In 1968, a relatively mild impact, only 1 million deaths, but half of those in people under 65 years. And this is generally considered 1968 as a mild pandemic, probably because one of the surface antigen was preserved from what was circulating before. So there was some residual immunity in the population. And then in 2009, we had a recent estimate of about 0.2 million deaths, so relatively mild also because, again, of some residual immunity in the population, with 61% death under 65 years old. And this is work actually done back there in the row by John and Lona and others. As a reference for contemporary seasonal epidemic, I mean, there's a lot of attention devoted to pandemic flu, but regular flu kills, more deaths if you take the cumulative impact over many, many years. So that's also something interesting to look at. And so on average, there's an estimate of 0.4 million deaths. That's also quite new, and this is work that's still ongoing, with about 1/3 of death in people under 65. So for pandemic, we have a large proportion of death in the younger population, but for seasonal, most of the deaths are in the elderly. So this is a review that we did a few years ago looking at historical data from London from the 1889-1892 pandemic, Copenhagen in 1918, the US for the 1957, and England and Wales from 1968, to illustrate the point that most of the impact of a pandemic doesn't necessarily happen the first year of circulation. It can be spread out over multiple waves of infection. And so, for instance, here in 1890, you have 3 recrudescent waves. This is London again, and so there's a relatively little impact. Only 10% of death occurred in the first winter of circulation. The second wave of the pandemic is actually out of season. Here in pink, you see the traditional winter period where flu is circulating, but the second wave of the 1890 pandemic didn't match that pattern. And then the third wave had relatively a large number of deaths, right? So if you had had a vaccine within the first year of circulation of that 1819 virus, you would still have prevented quite a few deaths. Copenhagen in 1918, so we had very few deaths in the summer of 1918, but a high infection rate. We'll get back to that. And then most of the death in the fall of 1918, which has been seen in a lot of countries around the world, and then some recrudescence in 1919 and even in 1920 in some settings around the world. There's actually relatively little that has been done on the 1957 pandemic, but in the US there's pretty good data, and again we see that the impact has been quite spread out over the first few years of circulation of that H2N2 virus. And then the last here on this graph is quite interesting. In 1968, what we see is that the pandemic impact was delayed to the second year of circulation in most of Europe and Australia, but not in the US. So there's also some geographic heterogeneity. But again, the take-home message is that not everything happened in the first year of circulation. Okay, so a little bit on the 1918 pandemic, clearly a widely studied pandemic because it's so catastrophic. And so this is data from Copenhagen where records are really interesting and detailed and go back very far in time, where already by 1910 there was a network of physicians reporting weekly on flu, measles, pertussis, and a bunch of other diseases that was affecting the community. And also, like a lot of European cities, they recorded deaths. So we can look at both morbidity and mortality there in this system. And we see that in Copenhagen, if we look at morbidity, which is the top graph, we have a summer, it's kind of hard to point sideways, but I'll just forget about it. So we have a summer wave here, which you see as quite a few illnesses, and also very unusual as compared with what you see in typical summers in Copenhagen, and then it's followed by an autumn wave and then a winter 1919 wave. If you look at respiratory death and you look at the summer wave, there's actually very few deaths occurring in this summer outbreak, and then most of the deaths are in the autumn of 1918. So we can look at the growth rate in weekly cases, which is something that we epidemiologists really like to look at, because it's a measure of how fast the epidemic is growing, how transmissible the virus is, and so how strong the intervention would need to be to try to mitigate the outbreak. And so one way to approach this is to look at the growth rate in weekly cases. And this is here on a log scale, both for the summer on the left and then for the fall on the right, for three cities in Scandinavia, Oslo, Copenhagen, and Gothenburg. And you can see from just the slope of those curve here, those straight lines, that the fall wave of the 1918 pandemic had a relatively low growth rate as compared with the summer. Right, so there's a high reproduction number, high transmissibility estimated from this data, just a quick growth rate of the epidemic in the summer. And so we can tabulate how many deaths, how many illnesses, and what's the ratio of those, the case fatalities, so measure of severity we have for those two waves. And we see that the case fatality rate was only about 0.3% in Copenhagen, but increased almost tenfold by the fall of 1918. And because we have seasons from around that time, we can see that this 0.3% in the summer is actually quite similar to what we see for seasonal flu in contemporary Copenhagen at the time. So what's unusual is really the fall wave of the 1918 pandemic with such a high case fatality rate. And as I said, the reproduction number is above 2 for Copenhagen in the summer and goes down to closer to 1 in the fall, which is consistent with a fraction of the population acquiring immunity from the summer wave. We can also look at the age distribution of death, and the age distribution of death for the 1918 pandemic is also something that people have really agonized over. But in Copenhagen, again, very clear data. So we can look here at the background death rate for a very detailed age group. I don't know if you can read the axis, but we have it down to month of death for small kids there, and then 10 years of age. Afterwards. And so in red, you see the background mortality in October, November, which is the month where the fall wave occurred of previous years. So it's a good baseline. And then in blue, you see what happened during the fall months of 1918. And then you see a sharp peak around 20 to 35 years of age here. And on the right, we can look at the relative risk of pandemic death, which is the blue divided by the red. So we're looking at how unusual the pandemic is in the different age group, and you see that it's very clearly unusual in the middle age groups. You can see here on this graph also that there's very little excess impact, very little departure between the blue and the red on both extremes of the age spectrum, both the very young children as well as the very old or seniors here. And we see that some of those distribution at the extremes actually vary between countries. So more recently, we've been interested in trying to document the timing of the pandemic a little bit better. So this is just a subset of a large table that we've built by just reviewing every paper that had been published on the 1918 pandemic, both historical but also more contemporary, to try to pinpoint when the peak of the pandemic was, but also whether there was evidence for a herald wave, so a wave of a mild nature that might have occurred before the month of September or October 1918, which is when the lethal pandemic wave occurred. The evidence here, of course, it's not virological evidence because there's very little virological information. So we're looking for epidemiological time series that would point to a risk, unusual deaths in young adults, which is really the telltale sign of this pandemic virus. And so by compiling this information, we can plot maps of the timing of the herald wave of the 1919 pandemic. So of course, there's a lot of white on this map, which means a location for which we have no data. And we hope that, as time goes, we can fill this out a little more. And then we can see in other countries different timings of epidemics, starting in late winter and early spring in Northern America, and then moving to April, May, June, July in Europe and other countries. We also see places where there's no evidence of herald wave, in particular in Latin America, in Peru and Chile, from analysis of epidemiological data. So it could be that the herald wave is so mild that we can actually not spot it on mortality records, so it's not necessarily definitive evidence, but at least it's not as marked as in the other countries for which we have data. And we can see also evidence of differences in timing at the scale of the US. This is here the East Coast of America. And we see that some of the earliest evidence here are from New York City, ship arriving into New York and carrying soldiers returning from Europe, and then spreading outwards in later months of 1918 on the eastern seaboard of the US. It's a very patchy map, right? I mean, ideally we would have data from many more locations, but it's just the evidence is not there. So in the next part of the talk, I'm going to touch on pandemic mortality burden. It's actually an area where there's been a lot of work recently, both on contemporary flu seasons, on the 2009 pandemic, but also trying to look back at historical data to try to get a better handle on how many people died. And measuring the impact of flu on population is really almost more an art than a science. You really have to have experience with data from a lot of countries to try to understand the patterns, and they're generally complicated. The main issue is that the clinical symptoms are not specific of flu, so you have to rely on broad outcomes, whether they're respiratory infection, all pneumonia as influenza, or even cardiorespiratory deaths. I mean, ideally, that we would have lab tests, we would have lots of confirmation, but it's just not happening on a regular basis anytime soon, and especially if you're interested in historical epidemics, there's going to be very little of that. So we have to rely on statistical methods to estimate excess mortality or excess morbidity above what you would see in traditional season where pandemic flu is not circulating, or when flu in general is not circulating. The other thing to think about is that during pandemic, as we've seen in that table earlier, we tend to have a younger age distribution of deaths as well as cases, but that's seen in death as well. And so if you just tabulate the number of deaths, you might not see a lot of them, but they happen in younger age group than typical. And so you might want to think of metrics like years of life lost instead of plain deaths to try to really quantify the burden of the pandemic and potentially how you want to allocate resources. And this is work that we did actually before the 2009 pandemic, but to try to think about this issue between deaths and years of life lost. So we have age groups on the x-axis, and then we're looking at both death rates but in red, but also years of life lost metric in blue. And you can see that when you look at years of life lost, the curve of burden is shifted towards the left-hand side of the curve. Of course, the young deaths weigh more in this metric, where you multiply the number of deaths by the life expectancy of different age groups. And in particular, in 1918, you see a large shift towards impact in younger population. And so once you have this metric, whether they're death or years of life lost, and maybe you have a limited intervention, for instance, you have just a few vaccine doses, then you can start to go down the age group and prioritize which age group you might want to vaccinate. So that's just looking at burden. Another decision you might want to make is also thinking about transmission dynamics and which age group might drive the impact of the disease. And traditionally, especially for pandemic flu, this tend to be younger population and school children. So that weighs even more towards trying to protect or control the outbreak in younger population. Okay, so in the spring of 2009, we had this really interesting pandemic event in Mexico City, and this is work led by Gerardo a few years ago, who had access to data from deaths from hospitalization records quite early on in the pandemic. So it was really one of the first epidemiological description of this pandemic published at the time, just a few months into the pandemic. And you can see in red here the age distribution of pneumonia deaths in spring of 2009, so in Mexico where the pandemic originated, versus background years in gray here and in black. And you can see this shift of deaths towards the Middle Ages here. For this 2009 pandemic. So already a telltale sign that the pandemic was quite unusual in the middle age groups. So a few months later, we were able to access the vital statistics from Mexico. And Mexico is particularly interesting in that respect because they publish their vital statistics very early and they make them publicly available online at the individual death certificate level. So that's a really good country, a really good sentinel country in a way to have. For instance, in the US, we have to wait for 3 or 4 years before we can get the full record. That's not the case in Mexico. So here we see pneumonia and influenza deaths, which is a rather specific outcome for influenza, time series at the monthly time scale for 3 age groups, the under 5, the 5 to 59, and the 60 and over. And the orange area on the right-hand side is the pandemic period. And we can fit baseline to these pneumonia and influenza deaths to try to get at what would be the expected impact on mortality if flu had not been circulating. And then we can look at the excess during the pandemic to see what's the burden of the 2009 pandemic. And just looking at this data without any model, you can see that in the middle graph here, for the middle age group, you get a very strong impact in the 5 to 59 years. And a relatively low impact in the under 5. In fact, there are many other seasons with more deaths, than in 2009, in particular the 2003 or 4 season, which was a plain flu winter outbreak, caused more death in young people. And then in the 65 and over, it's interesting, you do see a peak towards the end, the very end of this graph, but it's actually after there was any confirmation of flu from lab surveillance. It actually coincides with the RSV outbreak that they tend to have every year in Mexico, right? So we have a sparing of seniors here for Mexico in 2009. So we can look at a more detailed age distribution of death here. For Mexico, so in black you see the pandemic in 2009, so this is for the first year of circulation. And you see again the bump starting at age 15 and dropping down at around age 65, which coincides with probably a proportion of the population that had seen H1N1 back in the first H1N1 era from 1918 to 1957, an era in which the virus was close to the virus that came in 2009. And then for reference, you see 2 curves for seasonal epidemics, so severe H3N2 season, in 0304 in red. And interestingly, the severe 0304 season had more deaths than the pandemic in Mexico, right? But just the age pattern are widely different, right, from the pandemic. And then just an average of seasonal flu with the traditional J shape of mortality here. So in the US, as I mentioned, we were in a worse situation data-wise in the sense that we didn't have vital statistics for quite a long time, so we had to rely on all sorts of extrapolations to try to get a handle on what the burden of this pandemic was. So this was an early paper where we look at a subsample of cities in the US, the 122 city surveillance, which is a system organized by the CDC. Where we could see that the mean age of death was quite low here, 37 years in 2009, versus above 60 years for the 68 and 57 pandemic. And just for reference here, I'm also showing for 1918, 27 years of age. Again, an age distribution of the population widely different in 1918 too, just people were younger. And then for comparison, for an H3 and 2 season in the surrounding period, the mean age of death is 80 years, right? So quite different from 2009. And so here we're making the argument that if you just look at plain death, which is the left-hand graph, the 2009 pandemic, which is in blue, doesn't look very impressive. There's just very few deaths, right, especially as compared, and the different shades of blue are because we're using different extrapolation models. And then if you look at H3 and 2 seasons, or the 68 or the 57 pandemic, they have many more deaths. However, if you shift to years of life loss metric, the 2009 pandemic, because it has such a young age distribution of death, then takes over, at least for some of the season. The 1918 pandemic is not even on this scale because the death, it would just be out of that graph, just the amount of death was just too high. So a few years later, I mean, there was a very strong and systematic effort to quantify the burden of the pandemic in different countries, and this is work led by John and Lone in the back here. And so, but illustrating very interesting variation by geography in the mortality impact of this pandemic. So here, just for illustration, we're looking at Mexico on the left, which is probably one of the most severely hit countries for this pandemic, which is that we have data for. So the red is the impact of the 2009 pandemic towards the right-hand side of the graph, and then the blue are seasonal flu epidemics. And you see 3 different age groups, so all age at the top, the kids or people under 65 in the middle, and seniors at the bottom. Right, so as we've seen before, the red area is concentrated in the under 65, and there's very little impact in seniors in Mexico. But if you look at France as comparison, well, you really struggle to find any impact, right? There's very little red on this graph. If you really squint in the under 65, you see a little bit of red there. But so overall, the death rate is estimated at 1 per 100,000 for France for the pandemic, but 13 per 100,000 for Mexico. Right, and so John and Lone and many collaborators did this for 20 countries in which they apply systematically the same modeling approach, being very picky about the modeling approach, right? But just to try to be really systematic in different countries. And you see here on this map that in general, Europe was mildly affected by the pandemic in terms of mortality. There's sort of an average impact in Eastern Europe. But most interestingly, the Americas were quite hardly hit, and in particular, Argentina and Mexico had quite a high mortality burden there. And so then you can extrapolate this to the world population using models that are driven by socioeconomic indicators, latitude, and other things. And you can see, again, a large impact of the pandemic in the Americas. So there's a strong regional effect, and there's also a socioeconomic effect. So countries that tend to have lower socioeconomic status tend to have higher death rate. And so the estimate there based on respiratory death is for about less than 200,000 deaths, but really a great majority of those deaths in people under 65 years of age. So more recently, we've been working, and again with John and Lone, on trying to extend this approach to seasonal influenza. It's nice to know it for pandemic, but we would also like to know it for seasonal flu, especially as we're ramping up vaccination in different countries, and we always have new strains coming in, so we would like to monitor the impact of flu over time. And so this is work where we've gathered data from 33 countries with great help from the CDC, and that covers actually 57% of the world population. Those are the countries on the top map. Again, a lot of low-income countries are underrepresented, right? We have very little from Africa and relatively little from Southeast Asia as well. But it's a good chunk of the world population that we have data for. And we apply a similar approach to those 33 countries to get excess mortality rate for that population that we have data for, and then extrapolate globally. And when we do this, we see that we have an impact of about 4,000 deaths, which depends on exactly on the year and some of the assumptions made, but it's around 400,000 deaths. And 66% are in people over 65 years old. So it's the reverse of the 2009 pandemic, right? 2/3 in over 65. So recently, we've also been interested in taking a look back at the 1957 pandemic, which is sort of not exactly a forgotten pandemic, but it's sort of before the age of digital vital records. And also not 1918 in terms of severity, so people haven't really looked back at it that much. But we looked at a WHO database that has all-cause mortality by fine age groups for a bunch of countries. And here we see about 40 countries for which we have annual records of mortality. This is here respiratory deaths, right? So it's relatively specific. And from 1950 to 1970, so a good chunk of time where we can look at before the pandemic and after the pandemic. And so the red shows high mortality and the yellow shows low mortality. And on the left, we have all age, which is sort of a relatively mixed pattern of mortality. But if you look at the right graph, which is the 15 to 24 years old, so like a young age group that was relatively affected by this pandemic, you see that around the time of the pandemic, so 57 to 59, and in particular in 57, we have a high mortality impact. And because we have the surrounding years, we can do excess methods where we took out the surrounding years as baseline to look at excess mortality. And so, again, we can look at excess mortality rates for countries for which we have data, which is the top graph. And interestingly, we also see that Latin America has quite a high impact for this 1957 pandemic. And then we can extrapolate to the world population using the same type of socioeconomic indicators and latitude and so on. And we see that we have about 1 million excess deaths attributed to this pandemic in 1957. So a death rate of 4 per 100,000, so much higher than in 2009. And then this is work done much earlier, and that was discussed also earlier today, but quite interesting also to try to gather data from as many countries as possible to look at geographic heterogeneity in disease rate, and showing this large difference from 0.2% death rate in Denmark to 8% in India. And I think a really interesting project would be to try to go back to some of those data sets, right? Especially the countries that look like they're very hardly hit from this graph. India is one of them, but it's not the only one, and try to get at more refined data, because this was using quite a crude method. But this is a very large range of heterogeneity, right, in excess mortality rate, and it would be nice to know what's behind that. Okay, in the next few slides, we're going to shift gears, leave disease burden models, and talk about transmission dynamics and transmission models. So as I was alluding to earlier, one of the things we're really interested in quantifying is the reproduction number of epidemics, because that tells us how transmissible a pathogen is. So we're interested in comparing different seasons. We're interested in when there's a new virus coming in, seeing whether transmissibility decreases over time as population immunity builds up, for instance. And so here, one of the earliest work that we did was to look at historical data from England and Wales, where in an article in the 1970s, they had really nicely computerized weekly death rates from influenza, so it was flu-specific deaths, for a bunch of severe seasons in England and Wales. So you can see the 3 waves of the pandemic, of the 1918 pandemic on the left of this graph here. You can see the 57 and 68 pandemic on the right-hand side of this graph. And then a number of severe flu seasons in black in the middle. And then an epidemic in blue, 1951, that we were particularly interested in because from other data sets that we have from both England and as well as Canada, it looked to be a very severe season. Actually more severe than the 1918 pandemic in some locations. So we were interested in looking at that and trying to see whether transmissibility was particularly high for this season. And at the bottom here, you see an estimate of the reproduction number based on how fast the number of cases rise in those different epidemics. And you can see that in the 1918 pandemic, we have a high transmissibility between 1.5 to 2, and that's also true of 57 and 69. But we also have a relatively high transmissibility in the 50-51 epidemic at the time, especially in this data in England and Wales, transmissibility that's above 2. So at the time, actually, the WHO surveillance network was already in place, and they were monitoring flu, and they couldn't find anything different in the virus based on the surface antigen. Antigenically, it seemed a virus similar to what the populations had seen the year before. But by going back to the genetics of the virus and doing phylogenetic analysis, we were able to show that the external genes didn't change, but the internal gene actually changed. There was probably a sweep with a new polymerase gene, which helps the virus replicate. And so perhaps it's consistent with a feature polymerase gene that translates into higher transmissibility in this population. So higher transmissibility is not necessarily associated with higher susceptibility, right? It could also be associated with other changes in the virus. So this is an old outbreak, right? There's not a ton of data there. It's quite hypothetical, but it's something to consider. Again, we're very worried about pandemics, but seasonal flu can be, sometimes as severe and perhaps as interesting as pandemic flu. Okay, so we've we and others have been very interested in looking at the impact of schools on the transmission dynamics of pandemic flu. And this is work led by Gerardo a few years ago, looking at data from Mexico. So again, Mexico was one of the first hit countries, and so they were really worried about the situation there and the pandemic, especially as there was not a lot of information available, and people were scared that maybe this was another 1918-like pandemic. So here we see the log of the daily number of H1N1 cases, lab-confirmed cases during the pandemic. So starting in late March 2009. And there's a period where there's a natural spring break that occurs. So the schools are closed for the spring in Mexico. And we see that the reproduction number is 1.3 here from those data. And then the schools open, and the reproduction number jumped to 2.2. And then that's when the pandemic hit the news and everyone got worried, and so the Mexican government decided to close schools as well as restaurants and other gathering for an 18-day period. And there we can estimate that the reproduction number fell to 1, meaning that there's, one case will generate only one secondary case, so there's just a stable incidence. You don't actually have an epidemic. So we see that based on that, we can fit models to this data, and we see that the school closure reduced transmission in Mexico by about 1/3. And in parallel, there was other work done in Hong Kong showing a very similar impact of closing schools. In contrast, Chile and Peru, they did try to close school as well, but they did it too late, and we can show also using modeling that there's no impact. If you do it too late, it's just not worth doing it. If you're going to do it, you have to do it early. So we've been also really interested in understanding the spatial diffusion of flu. What are the drivers of flu diffusion? How fast does it spread? Does it differ between seasonal and pandemics? And we've been lucky to get access to a very interesting database of medical claims form that are sent by physicians to medical insurance. and that's for the US. So the US is a really nice chunk of land. If you're interested in spatial diffusion, it has a large span of population sizes and climates as well. So it's a range of connectivity. So it's an interesting location to look at. So based on these medical claims, we have records of flu-like illness for 77% of the US population. So it's quite a large population. And so we can, of course, a couple of cities are too small, right, to see anything. So once we've excluded those, we are left with 336 cities that report ILI on a weekly basis. And they're the cities here in red on this graph. And we are here capturing flu-like illness, which is a combination of flu or fever with respiratory symptom or febrile illness with viral evidence. And we can also link those cities with zip codes and census, which allows us then to look at environmental data and statistics on workflow and air traffic for those cities, and try to understand how the disease spreads between cities. This is just an example of some of those time series. So we have that for about 8 seasons in the US. Again, I said 336 cities here. We're just looking at a subset of 4 of those cities. From the very small ones at the top, Spencer, Iowa, Bryan, Texas, down to pretty big cities at the bottom, Denver, Colorado, just to show you that these are pretty well-defined time series with sharp peak in winter, as you would expect, because this is flu. And so we can look at the pandemic, which is the last season on record here, shaded in yellow, and we can also look at another season of interest, 2003-2004, which is the one on the left here, which is not a pandemic, but it's an antigenic change in the virus, so sort of a mini pandemic that we can also look at. So the first thing we did was to try to make sure that those data were safe and in the sense that they actually captured flu. So we matched them with traditional surveillance records from the CDC, so lab-confirmed flu cases, and as well as also really good data that Don had for New York City, where there's been long-term surveillance of emergency department visits for ILI, which is traditionally a very good indicator of flu. So at the top here, you see the medical claims time series. The pandemic is towards the end of the graph there. And you see, well, you don't see, but you can imagine 2 waves. I mean, the sharp one is the peak here in the spring of 2009, and the second is the actual fall wave of the 2009 pandemic. And if you look at the middle graph here, which is the emergency department record, that's the gold standard, and they're very similar. If you look at the weekly correlation in incidence, that's the bottom graph here on the left, it's 0.86, right? So the two records, data sets are very well correlated. If we look at correlations in peak week or excess burden, we also see very good correlation between the 2 data sets. And as a side note, at the time that we were doing this, there was also a lot of interest in using digital surveillance and Google flu trends to look at flu and monitor the incidence of flu. And in New York City, it actually failed to see the first wave of the pandemic. But medical claims could actually see it very well. So in this data set, again, it's very nice because you can disaggregate it spatially at a quite refined time scale. These are just cities here within New York State. So we have about 20 or so cities within New York State, and just for the 2009 pandemic, so from May 2009 to February 2010, and you can see a lot of heterogeneity here in the patterns in different locations. For instance, if you look at upstate New York, such as Buffalo or Elmira, you see that the main period of pandemic activity starts in September of 2009. If you look at the main cities, so Queens, Brooklyn, Bronx, the main activity of the pandemic is in the spring of 2009. So asynchrony in the timing of the pandemic in those different locations. So what we looked at here, because the spring wave was actually quite dominant in New York City, But it was only dominant in a few cities in the US. So to look at spatial diffusion, it was a little sparse because there just was not enough cities that were hit by that wave. We looked at the fall of the pandemic, which was much more widespread across the US. And so here you see a map of onset, or timings of onset of the pandemic in those different locations in the US. And you see the scale here at the bottom right. Green and yellow means very early onset, while blue means late onset of the pandemic. Right? And you can see that the earliest onset occur in the Alabama, Georgia region, so maybe not everyone is familiar where this is, but this is where the epicenter of the epidemic here is in New York. And then you can see that it's sort of radially diffusive out of that. And then it takes about a 3-month period to reach the northern part of the US. There's also a jump here from the East Coast into Central California, and then diffusing out of Central California to the West Coast. So the first thing to notice here is that there's a strong spatial structure in this timing of onset, right? So that's what epidemiologists like to see, because that tells them that there's a strong diffusion wave here of the pandemic. It's also much slower than all of the models would have expected. At the time, we had lots of simulation models for pandemic flu, and they expected that the pandemic should just sweep the entire US within 3 to 4 weeks. And this is a 3-month period, right? It's the idea that when you have a new strain invading a susceptible population, it should just go very fast. But it doesn't, right? It just takes its merry time to spread throughout the US. So we can try to understand why this happens. And so one of the hypotheses at the time that was put forward was the role of schools. In late summer, the schools are still closed in the US, and there's sort of a staggered opening of the school, right? It starts to open early in Georgia, Atlanta, and then it sort of rolls up to the north of the US for historical reasons. But so we can fit models that have schools in them to see if the just school timing can explain the onset of the pandemic. So what you see here on this graph, the red are the observed data. So here it's we're lining up all of the onsets from earliest on the left to latest on the right. And then the gray lines are the fit of a model to the data when we assume that there is only schools involved here that explain timing. So basically, whenever you open school, the pandemic is free to spread in your population. And you can see that the model doesn't actually fit the data particularly well. It helps explain the beginning of the outbreak when it's so early in the southeast of the US. But then by mid-September, all of the schools are open everywhere in the US, and the pandemic still crawls right to the northeast and to the northwest of the US. So that's not enough to explain the spatial diffusion pattern here. If we do a model that has space in the sense that it has a strong geographic diffusion wave, your risk of getting infected is proportional to how close you are to an infected city. Well, it explains the data much better than the model with school. And then we can play with different, and we also tried specific humidity and other environmental factors. And then you can try to combine factors. And by doing this, we see that the best model is a model that has both schools and space, but the main effect is really space. And so school terms actually only play a minor role in explaining the patterns, only at the very beginning of the outbreak, but not for the majority of it. And again, we see that it's slower and more spatial than expected. And we think that perhaps here what's happening is that because we have more children affected during a pandemic, and children might be less mobile than adults. I mean, they typically go from home to school, but they don't go very far, while working adults might travel more. And so perhaps that's why we see such a slowly diffusing pattern of pandemic spread. So we've been taking this work a little bit further recently because there's still a lot of interest in understanding what drives the spread of flu, both pandemic and seasonal, at different scales. And it's been very well shown, both from epidemiological and genetic sequence data, that for between country spread, air travel is the main driver. And really, what else could spread the disease? You know, between the Americas and Europe, you have to travel by air. It makes a lot of sense. But at the scale of a country like the US, it's much less well understood. So here we have pictures of the network of air travel in the US, which has a very hub-like structure, right? You have very big airports like Atlanta, Chicago, LA, that really drives the network. And then all of the other cities are sort of connected to those big hubs. But it's a very hub-like pattern. And at the bottom, we see the network of work commutes, so where people live and where they go to work. And that's a much more spatially distributed network, where you tend to work close to where you live generally, right? Although there are some long-range population movement through work. I mean, this is based on census, and some people report living in New York City and work in LA. So they probably don't commute every day, but there is, you know, it's an illustration that there are long-range travel within that work commute network. But the basis of it is really the slow distance travel. So we can fit some of those different connectivity or mobility indices to the data. Earlier on, I showed you data for the 2019 pandemic, but we have also 7 other seasons that we can play with. And we can look at the fit of different models with this data. And so a measure that we like to look at is the Akai key. The lower is the better. And you can see here that the model that on the right-hand side that has air traffic does very poorly. It has a high Akai key number here with this data. And the model that does the best generally in almost all seasons is the model that's purely based on distance. And then the work commute model does sort of intermediate, except for one season where it does a little better than distance, but it's generally less parsimonious than the model based on distance. And this is interesting because up to that point, a lot of people have been claiming that air travel drives the spread of flu in the US, right? But here we can see that it's actually much more spatial than you would expect if it was driven by air travel, because you see this strong spatial diffusion wave, both of seasonal and pandemic flu. Okay, so I don't know how I'm doing with time, but good, yeah. So to wrap up, I'm gonna talk a little bit about seasonality, which is also an area of very deep interest if you're, I guess, a flu epidemiologist. And so this is going back here to work done a really long time ago when there wasn't a ton of data on flu, but there was a little bit of information from WHO FluNet, which is the global surveillance system for flu, which had a couple of years of data for countries in the Americas here, and we could at least do pie chart for how much viruses was isolated in different months of the year in those different countries. And you can see that in the northern part here in the US, you have strong circulation in the winter months, so December through February. In contrast, if you look at Argentina, it's also very seasonal, but it's focused on their winter month, which is May to September. And then in between those two extremes, we have patterns that are less defined, so less seasonal patterns of influenza. So this was using pretty scarce data set, but since we've been really active trying to get a more refined time series from a lot of countries to try to understand how seasonality varies geographically and what are the drivers of those. So here we had nice records from Brazil, really large country, large population size, very climatologically diverse. And we can look here at mortality caused by pneumonia and influenza, and in particular look at the peak timing of those in different states of Brazil. So here you have the 30 or so states of Brazil, and you see the timing of flu epidemics based on 15 seasons of data also. And you can see that the state at the top of Brazil, so which are in the northern hemisphere or in tropical latitudes, tend to have early outbreak that peak in April. And then as you go south in Brazil, you get to more temperate or subtropical zones of Brazil, and then you get later and later epidemics. So there's about a 3-month difference between the north and the south of Brazil. So we've been taking this work, and that's been done by Vladimir Alonso in our group, by looking at data from many more countries. So this is analysis of FluNet data again, but like 10 years later, with many more countries that are not contributing surveillance. And using those, mapping the timing of flu activity in all those countries. So you see here a scale on the x-axis from December to November of the year, and as a function of latitude. The blue box represents the northern hemisphere countries, and the red box represents the southern hemisphere countries. So right away, you can see that the high latitude countries above 23 degrees north and below 23 degrees south tend to have epidemics in their winter months, which had been well described earlier. It's nice here to confirm it with more data. And then in between those two latitudes, so between 23 north and 25 south, in those sort of middle latitude area, you see a very diverse timing of peak of influenza. So some of it might be driven by the rainy season, some of it may be driven by other things. But the nice thing is that this also affects the timing of vaccination, right? Traditionally, if you're a Northern Hemisphere country, you would use the Northern Hemisphere vaccine recommendation, which tells you to vaccinate in October. But you can see here that if you're, for instance, in Martinique here, you tend to have epidemics that are a little bit too late. Or if you are in French Guiana, you're in the Northern Hemisphere technically, but your epidemics tend to be more in the summer month. So you should tend to have use a Southern Hemisphere vaccine rather than a Northern Hemisphere vaccine. So there is clearly lots of differences in the timing of flu in different locations around the world, but can we explain why we see those differences? And here I think it's an area that's progressed quite a lot in the last 5-10 years, but we still don't have all the answers. So there was a lot of excitement about the possible role of specific humidity a few years ago, and this was sparked by nice experiments in animal models, in ferrets in particular, and guinea pigs, showing that specific humidity affects the spread of influenza through aerosol. So basically, if you have specific humidity is too high, and you doesn't transmit. And if it's very low, so if you're in a dry environment, then you tend to transmit more. And so then people became excited with this animal experiment and tried to find evidence for a similar relationship in epidemiological data. So we here looked at data from 80 locations around the world. So they were typically cities rather than countries, just to have a good handle on the environmental conditions here. And we recorded the peak timing of flu in those 80 or so locations. And you see those locations on the left-hand side of this graph. They're ranked here by the minimum specific humidity that you have in the different locations. So the dots are when flu is occurring in the location, and the vertical bars in gray measure how much fluctuation there is in specific humidity in the different locations. And you can see that in most of the countries here on the left-hand side of this graph, you tend to have flu at the minimum of specific humidity in each location. And then you get to countries that tend to have a high humidity year-round, and then you either get two peaks between the year, or at the very right-hand side of this graph, you see that those locations tend to have their flu in the high humidity months of the year, right? So there's sort of a shift depending on what your range of specific humidity is in your location. And this is summarized in this relationship here, modeled from the data, where we have the probability of an influenza peak as a function of specific humidity, which is high at both extremes of specific humidity, right? So typically, this here would be a pattern that Hong Kong, for instance, is observing with months of very high specific humidity, while here on the right-hand side, we will have Spain or the US, where there is a season where you have very low specific humidity, so flu will tend to concentrate on those months. So we can then extrapolate this relationship globally and produce maps of expected timing of flu activity. Which you see here. So in blue, we have winter peaks of flu activity, which coincide with the month of minimum specific humidity. So it's the left-hand part of the relationship that we just looked at. And then in green, you have peaks that tend to be during the month of high specific humidity, typically the rainy season. And we see that they tend to concentrate, obviously, in the tropical zone where we have those conditions of high specific humidity. You also note on this graph that there's a bunch of locations which tend to be white on this map, and those are locations where the model can't really tell which one is more probable than the other, and these are locations where it will be really interesting to get more data. There are also a couple of outliers in the models and locations where there's just no fluctuation in anything specific, humidity or even temperature. A place like Singapore, which is really stable year-round. And there we think that perhaps the timing of flu might be connected to population mobility from other locations that have seasonal outbreak. So it might not only be a local conditions, environmental condition, it might also be a factor of how connected you are and to which places you are connected that might drive flu timing. So now we're going to end with a study that's linking both seasonality and school timing, and that's in Mexico, which again is a really interesting case study because first it was quite severely hit by the pandemic and has good data, but also quite a nice climatic amplitude throughout the different states here in Brazil. And this is work done by James Tamerius a few years ago with Gerardo, looking at the asynchrony in the pandemic in Mexico. So the maps on the left-hand side represent the proportion of cases in the different waves of the pandemic. Mexico was well known for having three waves of the pandemic in 2001, one in the spring, one in the summer, and one in the fall. And you can see that the spring wave, the top graph, is really concentrated in a few states. It's actually, there's very few cases. The summer wave is concentrated in the southernmost part of Mexico, in particular Yucatan province, peninsula. And then the fall wave is more widespread across Mexico, but actually avoids the southern part of the country here. And you can see a summarized version of that here on the right-hand side, where you see case numbers for the southern part of Mexico, and you see that most of their activity is in the summer months, and then in blue for the central and northern part of the country, where most of the activity is after September, so more synchronous with what we saw in the US. So we can try to fit models to this data with different ingredients to try to see what best reproduces the patterns that we see. And some of the ingredients from what we've seen before are local environmental conditions, including specific humidity, as well as schools. And so this is what we see here on this graph. We have a reproduction number, a curve here on the left-hand side, which we take to vary with specific humidity, and then we can fit the relationship to this data. And we see that we get a sort of inverted J or relationship in which we have higher reproduction number at high levels of specific humidity, which typically are found in the southern part of Mexico in the summer. And this is what you see on the right-hand side graph here, just the average curve of specific humidity in 2009. It's just higher in southern Mexico than in northern Mexico. As expected, it's a tropical location. And so then we can estimate from the data what is the effective reproduction number, which takes into account this relationship of specific humidity, but also what is the whether schools are open or not. So the sharp drop that you see here in the data are when schools close. In particular, this is for the spring break. This is for the 18-day period when schools were closed in Mexico. And then we have another drop here because it's the summer break in Mexico, right? All the schools close and then they reopen in the fall. And we can see an estimated pattern of reproduction number in the south in red and in the north in blue. And what you can see is that in the first part of the year, especially in the summer month here, you see a higher effective reproduction number in the southern part of Mexico because of this slight advantage that they have of higher specific humidity. And then when the school closed in the summer, all of the reproduction number dropped below 1, so that stops the outbreak everywhere. And then in September, the outbreak starts again only in the north because the reproduction number is high enough that it can start. But in the south, they've already had a lot of cases, so there's just not enough susceptible to fuel another outbreak. Right, so here we have an interplay between local environmental condition and specific humidity, when schools are open or not, and how many susceptibles you have left in the population to fuel the outbreak. We also see in those data, we've played a little bit with spatial diffusion, and we see that a model that has the nearest neighbors affecting your own condition is the best model. And so that's also consistent with a very radially diffusive wave, similar to what we see in the US. So the spatial signature is also quite consistent, although we don't have the data at the same level of resolution. And this is my last slide here in conclusion. We've seen from looking at those couple of pandemics now that the occurrence is quite unpredictable. One wish one could predict, but we're really not quite there yet. But the impact, I think, is also quite unpredictable. I mean, we've seen those rather different pandemics, right, from 1918 to 2009. And we've learned in others, we've now looked back at 1889. It's also quite different from the other 4 that we have. So every pandemic is unique in a way. And there's been a lot of effort recently to try to get better estimates of the global burden of flu, both seasonal and pandemic, and especially to gather data from understudied areas in Africa and Asia, as well as South America. And I think more of that should be done because in the same way that every pandemic is different, in a way, every new country also reveals a different pattern. So it's really nice to look at the heterogeneity in a lot of places. There's been a lot of interest in understanding and describing seasonality, and specific humidity seems to explain some of the patterns. I don't think that's quite the answer. I think there's more stuff going on, and we need more information and more models there. But it is better than where we were 10 years ago. We see that school breaks are quite important for pandemic flu in a way that are not for seasonal. Right, just because the timing of the seasonal outbreaks is just later in the year, and so schools are not closed for the summer, for instance. So it doesn't affect timing as much, and also we have more cases in older population than for pandemic flu. We see this really slow and highly diffusive spatial wave of pandemics, which we really can't exactly explain. They're clearly not driven by fast air travel. They're driven by more local movements, but I think more work needs to be done there and trying to reconcile while a pandemic invading an entirely susceptible population should just crawl from city to city and not fly airplanes. And then I think there are still a number of unresolved issues that should keep us busy for, I think, quite a while, and including thinking more about the drivers of spatial spread and how that might vary for different viruses that have different antigenic novelty. Also interesting to think about prior immunity. This morning there's been some talk of priming and cross-subtype immunity that might affect some of the pattern here, as well as viral interference, both between different subtypes of flu, but also between flu and other pathogens, such as RSV, for instance. So there's been some work, some really beginning work on that, but it's quite interesting. And then a lot of work on the evolutionary pattern of the virus with more and more genetic sequences being produced and antigenic data as well, and refinement of the methods to analyze those. But really where we need to have more work is the link between those patterns and the population level impact, which I think is a quite complicated story because the immunity patterns are also quite complicated to understand. And this is my last slide, so thank you for your attention.